First, we have a presentation from Neuroscientific Biopharmaceuticals with the ticker code NSB, a company developing novel drug therapies for neurogenerative conditions. To tell us more, we are joined by Director of Operations, Dr. Alexandra Heaton. Good morning, Alex.
Hi, Jane. Thanks so much.
Let me bring up your presentation and we'll jump into it.
Okay, so Neuroscientific Biopharmaceuticals. My name's Alexandra Heaton. Thanks. So in my presentation, I plan to provide you with a corporate overview of NSB, talk about the target markets we're focused on, the mechanism of action of our drug, NSB, and then go into the very exciting in vitro and in vivo preclinical data we've gathered to date, and finally go through our R&D timelines. To give you a brief overview of our corporate structure, the company listed in an oversubscribed IPO in July 2018. Our market cap is currently sitting at 52 million. Our largest shareholders include Macrae Investment, which is the large private family office of Clough family, who are famous for Clough engineering, and a Swiss-based fund called Alpha Swiss. We're in a very strong financial position with 13.5 million cash on hand, and that's more than enough to see us through for the next 12 months. NSB's leadership team is a good mix of both the technical capabilities and capital market experience. The company was founded by Matt and Anton, who both have strong backgrounds in biotechnology commercialization. We recently had the good fortune of bringing on Paul Rennie as chairman, and Paul is the founder and current CEO of Paradigm Pharmaceuticals. He took this company from a biotech startup to one that's currently running a phase 3 clinical trial with a market cap of $500 million. NSB is a Perth-based drug development company developing targeted peptides with broad therapeutic application. Our lead indication in ophthalmology is glaucoma, and our lead indication in neurology is MS and Alzheimer's disease. But the potential applications extend beyond these two diseases, and there are other conditions that we may be able to impact as well. So what is our drug and how is it made? When the body encounters injury, we have an immune protein called metallothionein 2, which gets to work repairing the damage. Now, metallothionein is a very well-known pro-survival protein. There's only so much of it the body can make. So what we've done is we've isolated the most active site on this protein and copied it synthetically. And this is called a peptide, and our peptide is called NSB. Because it's manufactured synthetically, we've also been able to make the peptide more stable. And because it's based on the human immune system, there's very low risk of side effects. So what happens is NSB binds to these target receptors located on the surface of neurons and other cells. And when it does so, it causes a cascade of reactions, downstream signaling, which promote regeneration, decrease neuroinflammation, and increase cell neuroplasticity, a range of positive benefits that you'd want a drug to do if you had a neurodegenerative disease. So neurodegenerative diseases like Alzheimer's and multiple sclerosis have an enormous burden of disease. There's a huge unmet medical need, and we need drugs that actually stop the disease in its tracks. However, currently there are no treatments available which are able to restore damage done to neurons themselves. Next slide. We know from the work we've done in vitro that NSB is able to significantly promote neural survival and regeneration. We looked at cells from the cortex and hippocampus, as these are areas of the brain that are associated with memory. Also of note, we've been able to regenerate damaged neurons in the spinal cord, which is indicative of potential use in spinal cord injury. We've done efficacy studies in mice and shown that it significantly improved symptoms of Alzheimer's disease and slowed cognitive decline by more than 80%. And on top of that, we've also significantly decreased the amount of inflammation in the brain, which is attributed to the disease. In a multiple sclerosis model, we were able to significantly increase the amount of myelin and the number of myelin-forming cells. Interestingly, NSB was 30% more effective than the current FDA-approved drug called Copoxone, which had a peak annual sales of $4 billion. It also increased neural cell survival, which is excellent and of course what we were hoping to see. Our ophthalmology R&D is focused on preventing degenerative conditions of the optic nerve. Currently, over 5% of the population suffers vision loss due to damage of the optic nerve. And yet currently there are no drugs available to treat the optic nerve once it's become damaged. Our lead indication is glaucoma, which is the second leading cause of blindness in the world, and optic neuritis, which is damage to the optic nerve caused by an inflammatory response. Our R&D program in ophthalmology is built upon this foundational data in which we were able to demonstrate regenerative capacity of fully severed optic nerves in rats. And what we've got here is a picture of an optic nerve which has been cut. As you probably already know, normally when nerves are cut, they don't grow back, which is what you see in this first image in the untreated eye. However, when we treated these optic nerves with NSB's parent compound, metallothionein, we were able to show that the optic nerve regenerated by over 250% compared to the untreated eye. So this is what happened after one single dose, and you can imagine how excited we were. It made us want to keep researching it to see whether NSB would repair the nerves in other conditions affecting the optic nerve. Here we've got data specific to NSB in glaucoma, in a pig model of severe glaucoma, which is the closest experiment to replicate human glaucoma pathology. You can see in the images here, in the tissue sample taken from the untreated eye, on the right, the cells are highly disrupted and they've degenerated and died, whereas the retinal cells from the optic nerve are healthy and normal in the treated eye. This data demonstrates the disease-modifying potential of NSB in glaucoma. Additionally, we demonstrated that NSB is able to penetrate through the ocular tissue from the front of the eye to the back of the eye in tissue distribution studies. We've performed these tissue distribution studies over several time points and have been really impressed by how well the NSB product penetrates through the layers of the eye and how long it hangs around for. In our 14-day study, NSB was still detected in the ocular tissue after 2 weeks post-administration, indicating that patients will likely only need once-a-month dosing. We'll also be performing additional studies to examine the potential reformulation of NSB, also as an eye drop. The company is at a critical point in our development where we have spent the last couple of years dedicating our resources towards completion of the extensive INDenabling preclinical program, and have now reached a stage where we're ready to transition into human studies. We'll be releasing our 4-week safety data in primates soon, and this will be followed by IND safety data, which will allow us to commence our phase 1 trial as early as Q4 this year. So to summarize, NSB is developing a peptide-based drug called NSB with a broad range of applications, but most focused on MS, Alzheimer's disease, and glaucoma. To date, we've conducted numerous preclinical studies, both in vitro and in vivo, and have compelling data showing that it improves neural survival, decreases neuroinflammation, and has a host of other positive benefits. In addition to our compelling preclinical data, we've got comprehensive information on the drug's mechanism of action, and our program's supported by strong safety data. Our next steps are to commence our first phase 1 human trial while continuing to collect IND-enabling data. And I'll just end with saying this is a very high growth stock. We have multiple inflection points imminent. And thank you for listening to this presentation, and I hope this has excited you enough to get involved yourselves. Thanks so much.
Thank you for that, Alex. It was a great presentation. We've had a few questions come through, so I'll jump into them. So do you conduct your research in Australia or internationally?
We actually collaborate with some of the best research institutes around the world. So that's important because our research is all independently validated by these external providers. And so for the most part, these institutes are located in the US, some in the UK, and some in Australia. But when it comes to first in human trials, we'll be doing them in Perth, Western Australia, and then phase 2 likely around Australia as well in different cities.
Perfect. And just also, so how is the NSB drug administered?
So it's currently being formulated as an injection. Peptides are typically injected. That will be a subcutaneous injection for Alzheimer's disease and MS. However, when it comes to glaucoma, we're currently administering it as an injection into the eye, which is fairly common for other indications like macular degeneration is a very common injection into the eye, but we're also reformulating it into an eye drop because of our data, which has shown how well it penetrates through the layers of the eye.
Perfect. Well, thank you for joining us again, Alex, and we look forward to hearing further updates.
Thanks, Jane.